favorite shipwreck story is, or what's your favorite sea animal.
Up, you're off.
Oops.
There we go.
It's definitely a time when we can discuss the amazing flatfish for all those who are
Yes,
interested
just for the halibut again.
in them.
Joke's on repeat.
Guess what Jen studied in graduate school?
That's right.
We have been seeing a bit of fish down here. What kind of fish have we been
We've been seeing
observing?
Cuskills.
Yeah,
And we've been
Cuskills.
seeing
Cuskills.
grenadier. I think
Yeah.
both the abyssal grenadier and the,
Come on, buddy.
oh, Erica was showing them to me
Huh.
just the other day. I was trying to remember. There's a bunch of different species of grenadier.
Yeah, but I mean,
Do cuskills live in conduits like you'd
They just-
expect a eel to?
just get out if you want.
Do
to, I think.
cuskills live in conduits like you would expect an eel to?
Conduits?
Holes.
Oh,
No.
I've always seen them out and about.
Yeah.
Okay.
Cruising around.
Cruising around.
They're pretty broad, actually.
You mean like
Or
geographically?
vertically. No, their size
Oh.
at their head is pretty large.
And then their tail comes to this like a ribbon edge,
Yeah.
I'm
right?
in their
Yep.
video. It's good.
All right.
Hey Jennifer, one of our explorers would like to know what is a data logger and what is it for? Can you use it, please?
Wait.
Are you talking about the
Are you-
position on the ship, the data logger, or are we talking about the temperature probe or temperature data logger that we're trying to
I
put in
would.
place?
Like the
I-
I
human
think it
data
is.
logger or
Yeah.
machine data?
Yeah. Can you give us the cliff notes on data loggers?
Wait, so people
Try this
data
for a
loggers?
second, but I can probably get in there better.
Why don't we do
Oh.
both? We'll
Yeah,
start with the
sure.
people.
Okay.
Yeah, so, well, my job is, like right now we're taking the
Cool.
temperature reading, so I
Oh, yeah.
log it, I record it. I've got this little application here, it's called the event log, and I enter all the information in there, like what type of observation it is, any notes on the observations,
Nice.
what location we're at. My job is to take as detailed notes as possible. And
Go
also
wide, please.
make observations on what we're looking at. I grab screen capture, so I take images of any interesting features or just the ocean floor in general. Every time we sample, I take lots of images. And yeah, and then finally we summarize it all into a dive report that we
Ooh.
can then give to the scientists. So they can look back on all the dives
Video, can
and
you
they
push
have a
in a
good
little, Marcus?
summary of
Sorry,
everything
Marcus.
that we've done.
That's good.
Whoa, by Argus, there's the octopus in the
Yeah,
water.
in the water.
Argus cam.
Thank you for explaining that term of data logging. As far as the temperature loggers, those are just battery-operated sensors that we pre-program to sample the environment. So in this case, the temperature of the water every hour. And we've set it up to do that every hour for a year from 2 days ago. And so we deploy them on the ocean floor, and then when we're able to return to the site, we can collect it and download the data and get a good record of how the temperature varied or didn't vary over that time.
And the one that we're thinking of deploying has the red tape wrapped on it. You can see it on the porch there, right?
Yep, and you can see it in that that hose clamp is actually keeping that sensor itself attached to a T-handle, which is just to make it more visible, easier to handle.
Magda, thank you so much for that clarification. That was a bit of a brain vacation on my part.
No worries. Thanks, Scott. So just to confirm, that was about 5.1 degrees.
Right. I don't think we got it
Yeah,
quite in
which
the
is
same
very high.
spot.
Okay.
Are you going to try and hold it in a different way? Okay. We're just trying to get the probe into Hercules' manipulator arm in a different orientation, so we might be able to get that temperature probe farther into that crack in the rock.
Ooh,
Yeah.
little gymnastics.
She's very persistent.
She's just, I love how she's just hanging upside down. Like,
I mean, they brood upside down.
yeah, that's true.
Come on. Come on.
All right, can we put you on SPL?
There we go.
Pneumatic planking.
All right.
Should be better.
Hopefully.
Yep. Going up.
Yeah.
Not that much there.
I'm surprised
No,
it's not more
it's a better
rapid.
spot for it than that.
Many of you are noticing our hitchhikers aboard Hercules. They've been on for some time now, and they do appear to be just fine.
We're not seeing the temperature respond the way we would expect based on that other.
We are getting screen captures.
What about just to the left of the...
Well, this is going up.
Yeah, well, I had it somewhere then, yeah.
Oh, there it
hold
is.
that for a second.
Wow.
Oh,
That's a
yeah.
big
Oh, that's a
change.
big jump.
This
Why
is
don't
good
I
though,
see
because
it?
that'll.
I
Huh?
just refreshed my, like up here on the screen, I just refreshed it.
Oh,
And
yeah.
there's that jump, yeah.
But we're not getting anywhere near the 9. Maybe it'll keep going up.
Oh, yeah, it's 6.7 now.
And hers is a
Got
little
s-
ahead. So
Yeah,
we're
we got
7.2.
7.2.
Okay.
3.
So it's slowly rising. Looks like we're leveling out at 7.5.
Yeah.
So what was the temperatures you were getting where you deployed the other probes?
The other probe, we deployed one of them in a temperature spot of like 9.5, and then the other one was deployed in an area of 6.6. And they were with the, in the vicinity of those two temperature probes was a meter. It was very close.
Uh-huh. As he's moving it around, we're almost, right now at 7.9, 8.0. So, yeah, right under that anemone seems to be a little bit warmer. 8.2. What, we're at 9?
We were at 9.8 with the other sensor.
Mm-hmm.
Mm-hmm.
It's still climbing. 9.4. I think you got us pretty close back to that same spot.
Yeah, I think you're right. I think it's kind of tucked under the anemone a bit.
Uh-huh. Which actually is probably a nice landmark. That anemone hopefully will be, I mean, they can move sometimes a little bit, some anemones, but generally I think they're going to stay put.
What's that weird red stuff on the rock?
We're at
Over to the right.
9.6, yeah, I see that. I think this is looking pretty promising.
I think this is a...
Do you want me to
yeah,
dig
'cause
around in there a bit more? Are you happy with it?
I think that it, you know, just trying to get the probe right about where you are, it's not going to end up in the exact same spot.
Right.
But very close to this would be great.
And we're within the proximity of the octopus garden, correct?
Correct.
Okay. So this could be
Yeah,
the octopus garden probe.
confirmed. Yeah, Aaron confirmed we're just a little off
Okay.
from that ledge, but very close.
Then yeah, I think this is great. So then we have instrument loggers at all 3 of the sites that we knew were
Had
on this area.
And had been, yes, and that this particular location a year ago, in this general area, we knew we had, seeping happening. So it seems that it's a persistent flow here.
Yeah, right around in there.
And actually, maybe we could try to trigger that other squeezer.
Oh, it's a great idea. And we should take a Niskin here too before we leave.
Sure.
Okay.
Jennifer Brown, what is your position in the
watch
van
these.
right now?
I am Psi 2.
And Ann, what is your position?
I'm Psi 1,
Which makes you the,
second scientist. Psi 2 is the chief science leader. What's your name?
Huh?
What's your science
Watch lead.
Watch
Watch
lead.
lead.
not lead scientist?
Well, right now on this watch, I'm the watch lead. I'm not the lead scientist of the expedition. That's Chad King, and he's the watch lead for the 8 to 12 watch. Is that
Got
what
it.
you're asking? Am I answering the correct question?
Yes.
Okay.
There are just a bunch of people asking about the lead scientist. And then somebody just said, say lead scientist, I need candy.
Oh, lead
So
scientist.
there must be a school watching live.
Are we going
Are
for
they
the,
playing a deep sea science bingo or something?
Yeah.
What is the lead scientist to do in the lead scientist role?
You mean, well, what I'm
science.
doing right now?
science?
Yeah.
Are we
Yes.
going for the red handle here?
Yes, please.
Yes.
All right.
Wait, do you want to do the squeezer first or second? Does it matter?
Oh, yeah, that's a good point. Let's do the squeezer first so it's not in the way.
Roger.
And maybe give it 2 good college tries. This squeezer that we're going to try to use hadn't triggered at a previous site, and so
Okay.
we're not sure if it's...
That's number 3. That's the squeezer.
That box is just not in the
Yeah.
same shape we left it in.
I know. So I might have to like pick them up and look for them.
I did mention that when I said that's not as neat
not
as
how
we
we
left
left
it.
it.
Well, you could try grabbing it by its handle and then laying it on the deck.
The yellow.
Okay, you don't
Rope.
want me to try and pick it up in here?
I mean, that's up to you. I
Yeah.
don't want to make you feel
It's easier.
controlled.
Yeah, there's no guarantee that it won't be in the way when it gets put back.
Okay.
So if you're watching, as he takes this fluid sample,
Have it.
if we have a good view of the sampler itself, what you'll see is a little black plunger O-ring inside of the clear vicinity. So in that bottom part, and that
Yep.
top black O-ring is going to move up to the top, and the fluid samples is going to fill in
Script
between those
force
2
one
black
for you.
O-rings.
Always a treat.
Ashley, can you briefly tell us what you do in the control van?
Sure, so I'm just controlling the zoom and the focus and the iris of the Herxus main camera while the ROV pilots fly and position the ROVs and cameras.
So are you an engineer?
I'm the video engineer on this dive.
Again, if you're just joining us, I am Scott Simon. I'm from the Marine Science Institute at UCSB, and I am the Science Communication Fellow. My job is to join the scientists and the engineers in the van and
That is
watch
wonderful.
along and share with you guys what they're seeing and communicate your questions to them as well. They don't see your questions, only I do. Try to share the love here in the comm van.
Scott, aren't you the host of Saturday's Weekend Edition on NPR?
I am Scott, not the NPR, Simon. Thank you very much.
Okay.
And I've actually made my retirement for every nickel I've gotten
Yeah.
on that.
Yeah, I bet.
Aaron, what direction are we relative to that ledge where we first sit down at this site. Are we off to the?
Well, I have a target for Octopus Garden. That was what we were aiming for. But as far as where we first set down at this site, I can't guarantee they're the same thing.
Okay. Can we put a target here?
Yeah, for sure.
Okay.
I'm marking any place we do observations or sampling.
I was thinking like I was going to note here for this sample Octopus Garden, but then give it an orientation like west or east or north or south from.
Well, I mean, we those are all permanent targets in there, so you can deal with it. But whatever makes you happy, I can add to the notes.
I was just trying to figure out what to call it exactly.
Like, is this target called something like Squeezer 3?
It will be, yeah.
Oh,
Or
okay.
it will be what Magda tells me it is.
Okay,
It's
okay, got
sample
it.
number 13.
Sample 13.
So there'll be a marker drop for sample 13, and we can go back and see that, oh, that was Squeezer 3.
So folks, you can see in the bubble camera, is this around where you want it?
Yeah. Is that's right below the anemone, right? Kind of in that same hole that you were.
Yeah,
Yeah.
just over it.
And I don't think the funnel is going to be possible to use here because of the ledge.
Okay.
So we are doing this sampler without the funnel.
Can you note that Can you note that this was taken without the funnel?
Okay, yes.
Okay, am I okay to take it?
Yes. I think so. Yeah.
Yep.
Yeah.
Okay. Go ahead. Can you try to
Alright.
fire the sample?
No,
Can you write it?
I've already gone
Yeah,
to
I have
the
it here too.
grip force level. That should have fired that, and it hasn't.
So what
It's
will
just
we
giving
see?
it a little shake.
What will we see to know if it's fired?
You're going to see the pin is going to shoot out the back. So see if you look in the basket, you see some of
yeah.
them are triggered.
Okay, got it.
Yeah, not working though. No.
Okay, that's okay. Sometimes these things
They've
happen.
tried this one earlier
Yep.
too, right?
Yep.
Yep.
It was worth a second try.
Yep, for sure.
Okay, so no sample on that, but
No,
still
yeah.
trying a Niskin, yes?
Well, we'll put the red temperature
Okay.
probe here, and then we'll do a Niskin.
Jen, sorry if that answer was vague. It's just with our positioning and how deep we are in the ocean, I would call this whole thing Octopus
Okay,
Garden. Like,
no problem.
we're awfully close to...
Yeah, okay. I don't have a problem with that at all. It's just that I'd ask.
Mike, can you remind me what the glass beads are as part of the buoyancy system for the Hercules?
Oh, our foam pack is made up of.
And is
many,
that
many tiny
syntactic
glass
foam?
beads. Yes.
Ah, thank you.
Yes, it is.
Could have fired right then. He lets it go.
Yeah, I was kind of hoping it was going to happen.
Okay,
Okay.
so the logger now?
Yes, the red logger, please.
What's great is that the squeezers that we did get, the 5 that we got, they look full. They look like good, solid samples. We won't really know until we analyze the data.
I've yet to see one on the dive, but I know we've seen a lot of rockfish from time to time.
A lot of what?
Rockfish.
Oh, uh-huh.
Quite a lot of species of rockfish found in the waters of the East Pacific. I believe there's over 70. Dr. Milton Love is a premier ichthyologist or fish biologist that studies those rockfish.
Yeah, we are too deep for the rockfish. That's
Yeah.
why we will not be seeing them. There's much fewer fish species that inhabit the oceans at these depths. And we've mentioned a few though that we have seen and are likely to continue seeing the Cusk and the grenadier.
Yes.
Some people refer to grenadier as rat tails, I believe.
Yeah, I've heard that too.
And even there, we're deeper than a number
Okay,
of
can we
the.
zoom in on the
Oh, yeah.
Zeus? Yep.
or deeper than a number of the Grenadier species
good.
occur.
Yeah,
There we
that's
go.
good. That's the same place that you had the probe, right?
Yeah, so now I don't know. It's going to fall a bit, you know. when I-
Yeah, I would say it's
You
Closer
want
try
to
to get
the-
it in as far as you can.
just where I had our temp probe?
Yeah.
Mm-hmm. And I don't know how they, but if the handle ends up near that anemone, that
is
might
it
be
safe?
okay.
Can I push on it a fair bit? or-
Yes, you can push out. Just not, don't like.
Yeah. Blind here. Zoom in on the bubble camera.
Oh, yeah, sure.
Jeez. Guys are killing me. Get out of the way.
Just joined us right now. We're exploring with the ROV Hercules, trying to get some temperature readings,
Might be down a little bit too far.
other data,
Right.
the octopus garden,
It may end up falling there.
I
Davidson
know.
Sea.
Regardless.
I feel like it's not going to stay up. The handle is pretty long.
Oh, they might take it from you.
Yeah. What about? I wonder if we should.
My
It would
only
be better down into the right, even though it might not be as warm.
Yeah, one consideration here is that because this is an octopus active area, they could I mean, just by living, they might move the probe out of position anyway. So putting it where you're suggesting, where it's out of the way currently of an octopus, might be a wise idea.
You're
So-
talking about down here?
Yeah.
Yeah.
If we can get it to stay and at least when deploying it, not actively be just
Can we go full wide
leaning
for
on
a
an
minute video?
octopus.
See if I can smear this in my grip here.
Although we're seeing these images from the camera
Zoom in a little
aboard
bit.
Hercules,
It's good.
every time Hercules is down, we also have another ROV down, Argus, which provides two things, among others, light and stability for Hercules. Currently, the Hercules pilot is Mike.
Hey,
And-
Mike,
Yeah.
if you were to rotate your claw a 180 and so that the probe was just tilted up the other way.
How do you mean?
Yeah.
Oh,
And then could you lay it down kind of between those octopuses diagonally? So the tip is up towards the sea anemone and then the T handle itself is
Oh.
down the
oh
it's
yeah, I see what
downhill?
you want.
yeah, down the slope.
But you want to be able to recover it when you come back.
I bet those ROV pilots are going to
Oh,
be
yeah,
so
we'll
skilled.
Okay.
Figure something out.
But you're saying that would allow us to use this lower area to kind of support it, but would keep the probe closer to the warmth that we're
Yeah,
trying to
and
measure.
at least we might get touched by some of the octopuses, but it doesn't seem like anybody's actively occupying that area. So it's like a
And then like this, right?
yep.
Yeah. Lay it down in between the two.
Excuse me.
Oh, that might leave her there too. Attach.
Just
Yeah, so
there.
currently that's Mike operating, flying ROV Hercules, and we've got Jessica piloting Argus.
Pushing us away here.
Oh.
Oh no,
Whoa, she took it.
She took
that
it.
didn't take very long.
Hmm. Hmm.
Oh, the handle looks exposed still.
Yeah.
Yeah, I
What if we can get
I've-
just pushed over to the right?
don't know the dexterity of the manipulator arm, but if it's possible, maybe you could.
Yeah, I can
Try to
nudge it
touch
around.
the-
I just like I have this one in between the jaws here now. I don't want to. Out of there. There we go.
Just try to maybe move it to the right of the octopus a little so it's not in her, on her body.
Yeah.
And
Just want to
Yeah.
Wish we could use our hands.
And one of the reasons that we don't want to place the octopus on the probe is obviously so we're not bothering the octopus, but also because in the octopus, if they do move, they might dislodge the probe from where we position it. And so we wouldn't necessarily be capturing the data that we're hoping to capture at this area.
Anchored then.
Do you want me to zoom in on that probe?
Yeah, zoom in. For what we can see here.
She might not let us move it though.
Come wide. That's good. She's got it.
Do you want to take it out of there if you can grab it?
If he can, I'm afraid he's going to take her with her and then get another
Hanger
Get,
on her?
get more
Another Klingon.
camouflage for the arm.
Yeah.
I mean, it's...
Oh, that might keep it in place though.
Let's just get out of here.
Oh,
Oh boy.
that's an egg sac.
Sorry?
That's an egg sac that fell down there.
No, it looks like just a rock.
Is it?
Yeah, I can see it on the bubble cam. It's just a rock, it looks like.
I would say we probably,
No, the
have
probe is
positioned
kind
that-
of where you want it, I think.
yeah, and it's kind of held in place by those rocks below it.
Yeah. That was the plan.
So
Did we move our anemone?
the anemone is now at the bottom.
Okay.
Yeah.
But are we doing any kind of different annotation for deployments?
I'm just marking them as observations.
Okay.
Yeah. And for marker, I just have Octopus Garden.
So in the computer, when we want to go back and see where we deployed red temperature logger, is it going to be something we can easily search for?
Yeah, so I made a note that it's placed in the area where we recorded the highest, like the temperature reading with the TDO sensor.
Okay.
Near the Octopus Garden, so hopefully we can refine, because I know that, Aaron, I think you've dropped markers for it, but I don't know if they have any numbers or if they're associated with any kind of labeling. Or
No,
anything.
I put whatever notes in you guys ask for. The only thing we were
Okay.
very strict about labeling is samples.
Right.
Yeah.
And I haven't had any strict guidance on how we want to label deployment, so just
Yeah.
label it the
As an
temperature logger.
observation. Okay.
We full wide video?
Yeah.
Yeah, and
Like
I'd
the-
like
the
to,
red
once
temperature
this
logger.
clears a little bit, I'd like to get a nice
'Cause
clear
these-
shot of the orientation of the temperature probe and the surrounding rock before we go.
Do we want to do an Niskin now or later, or not at all?
Yeah, we can do that.
Thank you.
No. Do we want a Niskin?
241.
Or
You want to know? No.
Oh, do you want a
No.
Niskin?
Yeah, let's do that. Do we don't need to move for that?
No.
Not
Okay,
unless you want us to, no?
hold on. Let me just
Alright.
ask.
I think that was
Mike,
8.
you're not
Let me
coming
see.
through on the
We're
audio.
just having an internal conversation.
Okay.
I thought it was 8.
Good question.
I think so. You can confirm with Mogda. Hey, Data.
Yeah.
To confirm, we have a Niskin that's downed. Is it 8 or 9? Do you know?
Niskin, that's what, available?
We had one Niskin that was out of commission. Was it 8 or 9?
Oh, so 8 was fired already, so we have a sample for that. I don't know about 9.
Okay.
It's
I'll try 9. If
Well,
it doesn't go, that's the
Let me
problem.
ask just one question. Amanda and Erica, were you this close to your deployments when you fired the Niskins, or should we reposition in any way? Or does it not matter? Okay. Okay, this is fine. Thank you.
All right. Open the iris a little bit, please, video.
Okay.
I already got eyes on 8. Oh
Roger.
dear, sorry. And I have that annotated as the red logger and Herc heading 241.7. Anything else you want in there?
Temperature was 9.8 and oxygen 57.9. It's C and micromole, respectively.
What was the oxygen? I missed that.
57.9.
Yeah, one that's fired.
Thank you, Erin.
Yep.
All right, that's, jeez. 9 is blue. Oh, that's
Yeah.
easier.
9 is blue.
So is that 9?
Yeah,
Oh,
that
so
one's
it's
just,
alright.
that one's already fired.
Oh, I just got it. All right.
Okay. So just to confirm, we just fired Niskin number 9?
Correct.
Okay, thank you.
Okay, great. And then can we take a few more close-ups of the temperature probe? You sure see a lot of shimmer from here, where it is.
Okay, zoom in there, video.
Can you get some stills here?
Yes.
Wow. Lots
A lot of
shimmer.
shimmering, yeah. Okay, I've gotten some images. Do you want a little more zoomed out, or is this good?
But let's do maybe as zoomed out as we can be also, just to see
Yeah.
the probe and the surrounding area. Did you guys have any way to record bubble cam images?
No, we don't. Just
Alright.
Herc and Argus Zeus.
Roger. Great. Did you get some stills?
Yes,
okay.
I did. Thank you.
All right.
All happy?
Yes, so we've got the Niskin. We tried the squeeze sampler. We've got the the temperature logger in place. So, let me think if there's any.
Some quick housekeeping.
Yes.
So it was still 013 for the Niskin, or did you log that separately?
No, 013 for the Niskin.
Okay, so
yeah.
Niskin and
Video to
failed
come wide on
squeeze
ours,
sampler.
please.
Yeah.
Okay, thank you.
And so when we look at the
Thank you.
sample log, we're going to see that as like all
All in
kind of
the-
one thing.
Yeah.
Okay, together.
Mm-hmm.
So this is really cool. Now we're going to go explore parts of this feature that we have not explored before. Is that the plan?
Yeah.
That's right.
And continue to, of course, catch our PCP.
Correct.
Yeah. How are we doing on that, Dean?
Yeah, so we're coming up. We're about 2 minutes shy of 5 hours. At 49 minutes after the hour, we will be up to 180 liters.
Great.
Already in the shot, there's 37 off.
Oh, I was just counting them. You counted
Wow,
faster
isn't it?
than I did.
And that's the ones that we can see. A lot of them are probably behind ledges and stuff.
Right. All right. Can we just pan a little to the left? Just curious how. Oh wow, they just continue on here.
Wow.
Oh, cool.
Very
This doesn't
linear
happen to be
pattern.
along our transit line, does it?
Porch light on, Jess?
We are
yeah, it is.
close to our transect line.
Yep, that's correct.
Wow.
Wow.
They're just everywhere.
Yeah.
I love how small they look in Argus Cam. They're just little guys.
Yeah, this is
So what's the
reminiscent
range
of
and
the
bearing?
view that we
The
saw as
start
we
of
were
the line?
departing. And
Start
this
of the
might be
line
the
is way
same
down
section
there,
of wall.
but I think they just want to get online.
We're going to
That's
drive
true.
like
This is
towards
very-
it from here.
Yeah, yeah. So we have ship range and bearing. If we kind of made progress down as well, looking at bearing 250, 33 meters. You're very close to it though.
And Aaron, which line is that?
Which line is that? That is a good question. I'll tell you in a second. You are 12 meters off, bearing 238 as you're heading. Well, the way you're looking. All right, let me see if I can find out which one it is. It's pretty censored on.
I'm thinking it might be the one that goes from 9 to 10.
Looks like 4.
Huh?
But,
Oh.
well, that's our label. I don't know if that came
let's
through
see.
from your data.
1,
Sorry.
2, 3. Okay, so this is the one we have as 7 to 8, then I
1, 2,
think.
3, 4, 1, 2. I think, yeah, you're right.
Okay, so we'll be picking it up closer to 8. Then,
Yes, is that okay? And headed towards 8?
yeah.
Okay, that's the closer end.
Yep.
All right, let's figure out where we need to go. Mike, you are truly almost on it.
Okay.
So I don't know how much I really need to move the ship any closer.
So we're basically going to pick it up here, go, and then I'm thinking we'll go like that.
Oh, great. So this is,
Maybe 10 meters off right now.
yeah, so this will be,
Yeah, we can just lateral, like
Yeah.
move Argus parallel to the line.
Yep. All right, so I'm going to start
Well, we
moving
could go
us.
this way and go that way. I don't know. I'm trying to figure out.
213.
Roger.
I'm trying to figure out what's more
213.
interesting. This line next or that line next?
Let me just see how very close we are.
Wow. What are these giant sponges?
Oh, I would live in one of those if I was an octopus.
Hey, Jess, can we look down a little bit at the
The
exhaust
face.
of the
So go
pump
down
system?
and then over here.
This, is that what you're talking about? Dane?
Okay.
You are about 10
Yep.
meters off, so do you want to step it in first and then
That
down?
looks good,
Or
thanks.
just start trucking
No worries.
down?
Let me know when you
Can you see what I
it's
mean?
10 meters
are all
down
done
there.
with the outlet.
Just 10
I
meters.
think I can probably make it over there.
All right. We good to move then?
No.
Science, you ready to go?
Yeah, to head to the transect, yeah.
Yep, so we're going to head to that selected transect.
So that's off and down slope, right? Okay.
We've
It
decided
should be
if-
along the almost parallel to contour, at least for a while. Then it will be a bit upslope as we get to the end. I can give you a warning. What steps are you comfortable with?
So is this the start? Where's
It starts way down here. Hold on, I'll zoom out.
oh yeah, but
We're
where
gonna
do you
pick it
want
up
me
in the
right
middle.
now?
Oh, I want you on here.
Okay.
And I'm just going to start ship moving parallel to that line. Good to go?
Yep.
So we're starting on line 8.
It's a line that runs from, on here, from 7 to 8.
Okay.
But we're picking it up about 2/3 of the way towards 8.
Okay.
So the first 50 meters will be along contour more or less, but then we will move up slope, but it's going to be less steep.
Sounds good.
All right.
are we, so we are on the line now?
We're getting there.
Okay. Are we facing in the direction of the line?
No, negative.
Not yet.
Okay.
We will
Oh,
be.
look
Now,
at-
do you want to run the lines like that, just facing on the bearing? Because it'll be along the slope, not looking like normal to it. Or do either.
Well, I think whatever gives us the
Yeah,
best
or we
view
can just change
of
as
the
we go. Just
octopus.
direct me or whatever.
Yeah, I think we'll need to kind of see which gives us more coverage
Yeah.
of the area.
because part of this is just trying to get a general view of
Account.
what's here, right?
Mm-hmm.
Yeah. All right, you're going to move us in closer to the line, right?
I was not planning on it. I was just going to go parallel, but if we want
I
to
don't-
step in closer, we can do
If
that.
we want to get right
Yeah,
on the
we might
line, I
need
think
to.
we'll need to, yeah.
Okay, so then how about we do a first move directly to the west,
Yeah,
10, 15 meters?
yep. sounds
15?
sensible.
All right, and then we'll start moving down.
All right.
Look at that
Sounds good.
overhang.
Okay.
That is pretty
Bridge nav?
substantial.
What?
That
Okay.
overhang?
Yeah.
Yeah.
Want to go look under it?
Yeah.
Yes.
Can I have a 15-meter step bearing 270?
Generally, if we're going to say yes to looking at things.
Look at those conduits. I really love that the octopuses tend to find holes that are shaped just so perfectly for their little
Right? It looks
Yeah.
like they
Bodies.
almost dug them out.
Yeah, they're
so
Well, there
fitting
have
these
been
pits.
some that look like they may have been slowly worn away over time by the octopus.
I know, it really makes you wonder, like, how long. How
they-
old is Davidson seamount?
How old?
Is it, 9.8 million?
Yeah, it's something like that.
Yeah.
I don't remember. Let me see if it's on this. No.
About as far as I
it's
can
not on
get.
this, but-
Okay.
Come back over here.
Moves in. I don't know how successfully it's going, but it's in.
They're in their little octopods.
So what's interesting
It's one
is
of
the
those fish.
fine layer of detritus up here on the top, and
Mhm.
nothing but those blackened cobbles down on the edges on the flanks.
It really helps you find the octopus because there's such a color change.
Yeah, I think you're gonna need more than
I couldn't hear you.
10 or 15. I'd do
Um,
like
it helps
40
you find the
meters over here
octopus
almost.
sometimes from far away because
Okay.
you see that
40?
color
Got
change
it.
from
Right on
the
it.
brownish
Nav?
green
Right,
to
right.
black.
You don't want to parallel it.
Yep.
You'd rather be Argus on the line too.
Well, it doesn't. If it's on it, then it doesn't matter. We can go like wherever.
Stand by.
It's just always being dragged out like this. Is kind of.
All right.
So let's get you happily settled
Yeah.
and then move the other direction. So 40 meters, 270,
Sure. Try that.
bridge nav. Can I have a 40 meter step bearing 270?
There's a Cordana right in front of us.
A what?
Oh, no, just kidding. That's just a black coral.
Okay, yeah, you dialed that in 4 times, so
Thanks.
for reals.
Thank you.
Thank you.
Oh, I look up and to the right. What's moving up there?
What is that?
Is that a fish or?
Yeah.
Okay.
Cuskeel maybe.
Oh,
I can't reach it yet
okay.
though.
No, it's all
unfortunately.
right.
Yeah.
No, at first I thought it was something different. I guess it was just down in the crack and looked different than I was expecting.
Yeah,
Jen,
it looks like a cuskeel.
is there a way you want me to log these transects? Like just mention the points that we're at as we
Sure,
cross
maybe
Well, and
our
if we
video on that coral.
When we see something interesting, I'm going to want to drop a marker on that too, so
Okay.
we can always go back.
Okay.
So if we see some, like when we were out with
Yeah,
Mbari,
drop
we
that
saw an aggregation
Yeah.
of them surrounded by all these
Yeah.
sea stars, which was
Yeah.
a really different looking grouping. And so
Right.
we want to note that, be able to come back to it.
Okay.
Now or in the future.
Okay. Thanks for the zoom. You got
Science,
some
I
good
was
stills.
thinking when we actually are comfortably on that line, I'll drop a point there so you know where we started,
Perfect.
and then we'll get set up how you like it as far as height.
Okay.
So we can start moving.
That sounds good. And then along the way, when we see unusual things that are interesting, we're going to want to drop markers.
Sure.
Okay,
And
full wide.
I'll drop it if you don't. If you want something, just shout.
Okay.
I'll happily drop it.
And Erin, you said we're starting about 2/3 of the way.
Yeah, I think so. I'll do a zoom out and we can get a look at it while we have a ship move in, but it's going to take a little bit to get shifted over. So
Okay.
we have time to look. I know it's really hard to see the HYPACK screen. I can turn off the background map if you want. Let me do that so you can see a bit better.
Zoom in on this floaty thing.
Huh.
Ctenophore.
It is a
Oh, it's
ctenophore.
beautiful and it's
That
orange.
is
Yeah.
amazing.
That's
Wow.
mine. That's mine.
Oh
it's
my
uniform.
gosh.
Oh yeah, Erin,
Looks
that's
like
your
a
favorite
jewel.
color.
Yeah. Good memory. Watch, friends. So pretty.
That is beautiful.
All right. Can you guys see HYPACK? Can you see where we are?
You can see
Sort
it's
of.
faring so nicely in there.
Yes.
And so comb jellies don't have the stinging cells that your typical anemone or jellyfish will have. They have sticky cells. And that's how they capture their prey. And then the combs is what you see kind of giving out those rainbow lights. Those are little combs along lines along the sides of it that it uses for swimming.
And they're cilia, right? just bands of cilia.
Is that right? I don't know.
Yeah.
Okay.
These are the largest animals that can swim using cilia.
Wow.
It's beautiful. I got lots of pictures.
Great.
Thank you.
Yeah.
Oh,
that
wow.
is just stunning.
It's perfect fall colors.
Yeah, I was
Yes.
just thinking that.
That's what I want to be for Halloween.
Oh, that would be pretty.
There's a xenophyophore down to the bottom left, off screen now.
Okay. We
What
zoom
was that
out,
again,
A xenophon.
Amanda?
Okay.
It's called a xenophyophore. It's a gigantic protist.
Oh.
A gigantic protist.
Really?
Wow.
Oh, the dark brown. So in the middle of this, you're
Yeah.
getting it centered up now. Yep. So
Oh,
zoom
that.
in.
Thought that would be like a dead sponge.
Wow, that's a protist.
Guys, so that's a single-cell organism.
Yeah.
A single cell
That makes it
organism.
impressive.
That is
What is it
my-
called again?
Xenophyophore. X-E-N-O, oh boy, P-H-Y-O-P-H-O-R-E.
Wow.
Okay, I got it up to the PHYO and then I was just freak out.
Yeah, then I gave
Wow.
up.
It's in the early pages of the guide.
I will find it.
This is incredible.
A single cell. And how big was it on the lasers?
At least it was over 10
Yeah.
centimeters across.
Wow.
Here it is.
4 inches.
Upper left.
Oh yeah.
Are the lasers on?
Yeah, can we get one?
They're
with-
out of frame, I think.
Thank you. Yeah,
Just,
I'll take one image with the lasers.
Well,
And there we go.
Thank you.
so the lasers are how many centimeters apart?
10.
Okay, yeah, wow.
Very neat, thank you.
Yeah, thank you, Amanda.
We have a couple octopus that are out and moving about here, as well as a number of brooders. Can we zoom in on these octopus that are out and try and determine if they're male or female?
Yeah, the RV pilots are just
Yep,
switching.
okay.
Again, that orange thing was
Sorry,
a-
did I hear? Did you guys want to zoom on something?
Up and to the right, there's an octopus that's actively moving about. I was just gonna, while we're waiting for the
perhaps a male.
ship move, I thought we could take a look and see if these are any of these are males.
Yeah, which one you want to look at? The one to the lower right or the one to the
Yeah,
upper?
we can start the lower right.
Sure.
So Jessica is now
Mike,
piloting
you
Hercules.
got an eye on the sonar, and you're now upslope of Herc. Oh, well, no, the ship is, so you will be eventually. I have no idea where you are right now.
Sure thing.
Unfortunately, the arm that we need to see is on the other side of this octopus, but it might still reveal itself to us.
Arms up.
I just love how slowly these guys move.
All right, we've completed that 40-meter step. Do you guys
Oh, we can keep moving then.
have a good feel for where we are? So we're now, the ship is now upslope of Herc. So Herc, Argus is going to
Be a little
peel
upslope,
in.
too.
Maybe a little scary, not sure.
Yep.
Oh, there's
Sure thing.
We're
Let's
just
see.
good here for now.
Do you want to wait for it to settle in, or you want to start moving down?
I prefer to wait for it to settle for a
Okay.
minute so we can
Yep.
see where we sit.
No worries.
So while we're doing that, we can
Go ahead
zoom
and
in on this.
one guy. It's good.
looks like he's got a chopped arm.
Hmm.
He's missing a tip.
Yeah.
Mm-hmm.
That looks like arm 1, and then 2, and then a gap. So 1, 2, 3, 4, 5, 6, 7. So sometimes the males hide
Go ahead and
that
come a little
third
wide,
arm. So
please,
that's
video.
possible that's a male.
Good.
50-50.
I love it.
2, 3.
So Aaron, how close are we to the actual transect line?
Oh, very close.
Oh, I see it now.
Right
Okay.
on it.
The ROVs are on it?
Hercules is based on its VVL position. So can you see the HYPACK screen somewhere?
Yeah.
All right, so I'll zoom in. So the ship is now upslope. This is Hercules with the pointer, at least based on Doppler, which is more.
Can my video?
I trust the USBL is kind of all over the place.
Good.
There are the other 2 markers you see moving around. So the green one that jumps is Argus. The red one that jumps is Herc based on USBL. And then the one with the pointer is the DVL. And that's what I'm using to position it.
Okay.
So your Herc's on it. We're just waiting
Zoom in video.
for Argus to swing in.
That's
So
good.
we get a feel for where it is.
So
a little
the
wide.
transect line is the red line with the arrows
trying
on
to
it?
get the
Yes.
Okay, I
guys
was
in the
looking at
frame.
the green dotted lines.
It was green dotted. I made it red so that we could see it a
okay.
bit better.
Got it.
Yeah.
Okay, thank you.
No worries.
Aaron, is there any way that we can see on the quad what you were just explaining?
Oh, we did this last time. It becomes really hard to see, but Ashley might be able to do it.
I don't know if this gets you a better angle on this guy or not.
Can you zoom in? Kind of right next to his, under his left eyeball.
So in the past, the males have not been advertising that arm, so it makes me wonder if this is male because that third arm's tucked away.
Yeah, I'm not able to.
Because I can't see it. I can see 1 and 2 and all the others.
Thank you for trying that shot.
Sure. Go ahead and come wide their video. All right, I'll head over to that line there.
You are so on
I'm
the line.
on it there.
You're totally on
currently.
it.
Sweet, so this is the beginning of transit
Yep. Just
1, transit middle.
So I'm calling it Transect 7 to 8, based on your naming. Sound good?
Sounds good.
So the consensus is we want to do some, whatever gets us the best viz on this transect line.
Yeah, so I'm right now, it looks like we're moving to the left.
The ship is at the end of its move. It's just jiggling a bit.
Okay.
I'll start moving south if you want to think about height.
Yeah, well, we'll be going down slope now, right?
Yeah.
Yes.
Are we facing in the direction of that we're going to be moving?
Negative. So this, I can show you real quick. So this here would be just about the angle or the heading of our transect. But as Mike said, we are going to be moving down the slope, so you'll lose some visibility of the bottom.
Okay.
Can you
always
zoom out on
lateral.
HYPACK, Aaron?
Sure.
And by lateraling, we can go kind of to the slope here.
Okay, so it's gonna get steep enough. You'll see when we go forward that we're gonna lose a lot of visibility, and what we can do is just flip the vehicles around and just continue the line just going backwards.
Great.
So that we can get a much better visual, right? So we'll just go forward because it's pretty flat right here. And whenever it starts to change, we can just flip everything around and just carry on.
Sounds good.
So are we starting about halfway down?
I would say
This one?
2/3.
Two-thirds? Okay, I'll make note of that.
So closer to 8 than 7.
Okay.
What is that?
It's just a little polyp coral, and then it's got a black background behind it. I
Yeah.
thought maybe it was like a sea mushroom.
I wonder if that was a... Can we zoom in on that little black thing? I'm just curious, we'll see octopus cement
Sure.
in there.
Good video.
Because it looks like it's been worn away.
Yeah.
Wow.
Yeah, maybe.
It
Maybe.
Could be.
Definitely like a sizable hole for a little brooder.
But Not much shimmer coming out of that hole.
Yeah.
No, and that's one of the questions. Is the shimmer at all advantageous to this? Is or
Right.
just coincidental that they happen to find sediment-free surfaces where the shimmer is occurring?
Or is it just a nice little nook
Yes.
in the rocks that are it's protective?
Yeah.
Yep.
I can hear you, Chad.
You know you got your shots there. All right, go ahead and come line video. Great.
You're muted.
This is just so amazing. We're beginning a transect here along. and
Chad, can you hear me?
Got a couple undergrads from UCSB,
it'll
interns
it would
at the
take-
Reef,
Aaron did the
Sam E,
calculations, it would
Jessica
take quite a
S,
long time.
Lauren W,
Like
and
over an
Madigan
hour
down
to
at
get
the
to
Reef.
an end point.
The most
So we thought
interesting
we might
thing
as well
I've
just
seen
be on
so
transect when
far
we get to
is
an end point.
everything.
So we don't
Oh,
lose
sorry, I
that
can't
hour.
actually hear science in the background. Can you speak up a bit into the
Sorry.
mic? No, you're good.
We are on it now. We're on 7-8.
Just having a conversation with Chad. They're
We
just
are
trying to
on
sort out
7 and 8.
whether or not we should go to the
2/3
start
towards
point
8.
of the transect or pick it up midline.
Roger,
Here.
understood.
Is this plan okay with you? Okay, great. So we're going to move from this point to 8, and then we'll be able to pick up the next transect line at its beginning.
Does he know which direction you'd want us to go to?
Does that sound good? All right. Yep, we're good to go.
Pilot's good to go.
Yeah, I would track a line, Aaron.
Check a line. All right, bearing 213.
213 at, I don't know, try 0.3.
Yeah, 2.3 would be nice.
Always speed it up if we have to.
What was the last bit? Sorry.
0.3.
23?
0.3.
0.3. Okay. Thank you.
23.
Not 20.
23. Jeez, oh man. All right. Yep.
I don't think I'd be able to make that.
Bridge nav.
I need it.
Should we reverse the...
Can we
Direction we're
track a
facing?
line bearing 213, 0.3 knots? You okay? Okay.
So should we turn to face not down into the abyss, but somewhere where we might be able to see a few octopus?
Sure, you guys want to face, so I'm doing perpendicular to the slope right now. I can face up slope if you'd like and back down.
Yeah, almost as if we follow the line, like Mike suggested, backwards.
Right, okay, so I'm going to probably keep around this so I can see a little bit what's coming ahead of me, if this works for you guys.
Yes.
Yeah, this is good.
Great, thanks.
Aaron, as we do these transits, does the ship move with us?
Yep. So that's what I'm calling in. I'm calling ship moves. The ship move controls Argus, and
Okay.
Argus controls how far Herc can go because of the 40 meters tether. So it all has to
Yeah.
kind of
and
happen together.
Over 3 kilometers, there's a lot of delay between moving the ship and seeing that like affected in Argus. So it's kind of a slow to get started on a line, but then once it's going, it just truck along.
But difficult to stop, right?
Yeah.
Yeah. So we're waiting for that move right now?
Yeah, that's
Can we
correct.
just do a little look around then? Just see what's behind us? And
Sure thing. Let me come over a little bit here. I'm speaking out loud too much now, I think.
Got some third graders from Forks, Washington that just reconnected, just finished their lunch and recess, and the students are wondering what octopus cement is.
Oh, yeah.
yeah, so that's the octopus when she lays her eggs, she wants them to stay put on the rock, so she secretes a little substance that cements them to the rock, so they don't move until they're ready to hatch, and then the hatchling will emerge, and the rest of the egg will stay behind, attached to the rock, until some animals come along and